It's really a pleasure to be here today to tell you about some of the work that we're interested in my lab. And I'll be talking today about the molecular and cellular mechanisms underlying the sensations of itch, touch, and pain, which, as Armand mentioned, I'm sure you've all experienced, perhaps on a regular basis to some extent. So our senses were first defined by Aristotle, who was really the first scientist and philosopher to try to describe how we interact with the outside world. He, for the first time, described the 5 exteroreceptive senses that allow us to perceive the outside world. And of course, I think maybe you've all learned this before in elementary school, that we have 5 senses: sight, smell, hearing, taste, and touch. But we know a lot about how we see the outside world and how our hearing works. We know a lot about smell and taste, but we know very little about our sense of touch. And to me, this is really surprising because touch is such an important part of our lives. And part of the reason is that maybe we take it for granted and don't really think about it so much. And also, it's been very difficult to study scientifically tractable. It was not very tractable. So, but over the last 10 to 15 years, there's been a lot of technical developments that have really started to reveal the mystery of touch. And perhaps I don't have to remind you, as we're all in quarantine and we're missing human contact, that touch is very important for emotional well-being. The bond between a parent and a child is first forged through touch. When we greet people, we shake hands under normal circumstances, we embrace one another. And I think we're really experiencing that lack of touch right now could be really debilitating. Touch is not only important for emotional well-being, but it's important for normal development. So premature babies who are in incubators have a regular schedule of tactile interactions with caregivers and family members. And we know that without that sense of touch and physical contact early in development, you could see a lot of emotional disorders as well as abnormal brain development. Touch is important for everyday tasks. So your ability to walk in a straight line without staring at your feet, our ability to learn to read Braille or play a piano concerto, and of course, the important task of texting, which we do all the time, are mediated by touch-sensitive neurons in our body and cells that constantly monitor limb position that allow us to enact coordinated movements. And also, altered tactile sensitivity is the #1 complaint in the clinic. And what do I mean by altered tactile sensitivity or touch sensitivity? There's a huge range of disorders that include tissue injury or disease that cause chronic pain in response to gentle touch sensations that don't normally cause pain. Loss of tactile sensitivity in the case of repetitive use injury where there's numbing and tingling. In the case of some forms of diabetic neuropathy, loss of tactile sensation leads to repetitive injuries that are the actual cause of amputations in some diabetic patients. And also chronic itch is a very common disorder that is equally debilitating as pain. And what I'll do today is tell you about some of our efforts to understand these diverse tactile disorders. And first, what I'll do is give you a brief introduction to how our pain and touch system works under normal conditions, as well as disease conditions, and then tell you a little bit about how my lab tries to unlock this mystery. So pain plays a very important role in our everyday lives as a warning system and is very important for triggering protective behaviors. Armand mentioned the touching a hot pan or a hot surface, perhaps when you're cooking. This helps us learn to avoid situations that are capable of causing us permanent harm. inflammatory pain in the case of a sunburn, or perhaps you had a sprained ankle, where normally things like the hot water in the shower or the gentle touch of your clothes against your skin or walking on your sprained ankle can cause pain. And this kind of pain is important because it tells us that we should take a break. We shouldn't use that twisted ankle, and allows us to heal. But chronic pain, unlike acute inflammatory or pain, serves no purpose. And somehow in certain people, in response to, for example, a minor sprain, in some people that hypersensitivity associated with that never goes away. And pain serves no purpose. And millions of people worldwide suffer from chronic pain. And while we often think about chronic pain as really debilitating, I just want to point out that the loss of pain or the inability to experience pain is equally debilitating. And here I'm just highlighting Ashlyn Blocker. There's a great New York Times article about her. When she was a baby, she never cried, even though she was constantly injuring herself, scratching at herself, causing self-injury, chewing on her fingers. And as she grew up, one instance that her parents describe is that one day when she was playing basketball, she was running around, she twisted her ankle and kept playing, but her family noticed that she had actually broken a bone and her bone was sticking through her leg and she was bleeding and had to go to the hospital, but she could not feel it. And when she was cooking, when she dropped the spoon into the boiling pot water, when she was stirring the pasta, she could just reach right in and get it. And she never experienced any pain associated with this, even though she was severely injured and had severe tissue injury. At the other end of the spectrum, Cameron Basdog suffered from a very terrible burning pain syndrome where small changes in body temperature from a brisk walk or a jog would cause severe burning sensations all over her body. And both of these conditions are equally debilitating. Chronic pain affects about 10 million Americans annually and millions of people worldwide. And there are no treatments or cures that significantly relieve suffering in most patients. And so these are very debilitating conditions that we really have no way to treat them. And so really the challenge that we and many other people are interested in understanding is how can we discover genes that underlie chronic pain so that we could develop new therapies and drugs. So what do we know about pain? Remarkably, some of the earliest studies about how pain is transmitted from the periphery to the brain came from Descartes back in 1664. And he remarkably drew, without any knowledge of nerves or how pain is transmitted, this sort of cord going between the periphery when the foot is in the fire and feels the heat, all the way to the brain, and described a mode of pain transmission from a heat source to the brain that triggers a sensation. And he got that quite right. This is indeed how pain is transmitted, through a series of neurons. And but of course, it's a little bit more complex than this. We don't have just one nerve. We have many types of nerves. And what I'll do is just tell you a little bit about how the nervous system is organized, so I could tell you about some of the work that we've been doing. So acute pain is defined as a irritating and painful sensation that's triggered by stimuli capable of causing tissue damage. And this could be noxious heat or noxious cold or noxious pressure. And these sensations are mediated by the neurons and cells of the somatosensory system. So here in this schematic, You could see in green are all of these free nerve endings that make up the somatosensory system that allows you to discriminate and experience gentle touch as well as noxious pressure, temperature changes, and also responses to some chemicals. So if you ever were cooking and got squirted lemon in your eye, your somatosensory system is what gives you that painful sensation from that lemon juice. But also the somatosensory system not only brings in information from the outside world, but plays a key role in monitoring internal state and the inflammatory mediators or chemicals inside your body when there's tissue damage. And we have millions of these neurons that densely innervate the skin and the viscera throughout our body. In my lab, we study somatosensory neurons, or these sensory neurons, from human donors, as well as using mouse as a model system. And the mouse is really nice because here you could see in this image of a mouse where David Ginty's lab labeled all of the somatosensory neurons in green. You could see that these neurons densely innervate the skin as well as the viscera, and has a system very much like the human system in terms of organization and cell types. So we know that there are distinct classes of sensory neurons that respond to innocuous and noxious touch. And so, for example, there's different neurons that allow you to experience the brush of a feather or the prick of a pin. Oops. And the first step in your ability to detect a painful stimulus is the conversion of a physical stimulus into an electrical signal that happens at the level of the free nerve endings in your hand or throughout your body. And this is called the transduction step. The second step is the transmission of an electrical signal. So transduction converts a physical signal into an electrical signal that then travels from the periphery to the spinal cord. An important part of pain and touch is that there's a modulation of that signal that happens in the spinal cord and integration so that that signal can then go to various places in the body. And only when that signal reaches the brain do you actually experience the perception of that stimulus. So there are also protective reflexes that occur as soon as that signal gets to the spinal cord. So if you've ever grabbed a hot frying pan, you might notice that you dropped the pan and then felt that ouch sensation. And that's because these protective reflexes are quite rapid, and it takes a little bit longer for you to experience that sensation. It takes a little bit longer for that signal to reach the brain. And so common pain drugs target different steps. So local anesthetics can act at the level of the spinal cord to try to block that signal from getting to the brain. And antidepressants and anti-epileptic drugs that are commonly used to treat pain act on the brain. And because of these locations in the central nervous system that these drugs act, there are often a lot of side effects, including loss of cognitive abilities, effects on sleep, motor control, and make them not very desirable. In addition, as you know, with the opioid crisis, that also leads to a lot of problems with addiction and also tolerance, where over time they lose their ability to alleviate pain, and more and higher doses of these drugs are needed. So we in my lab are very interested in trying to identify new pain targets in the periphery that mediate pain transduction so that we could stop pain at its source and not use drugs or therapies that affect the central nervous system, where all of these side effects and addiction really occurs. And what I thought I would do tonight is just tell you about some of the different approaches that we use to try to identify new pain targets in my lab as well as in our field. So the first approach that I'd like to tell you about is the use of natural plant products that target somatosensory neurons. And a lot of basic research has been done looking at natural remedies, as well as plants that are known to trigger pain or itch. And through basic science, we have now some of the most exciting new drug targets that are being pursued in pharmaceutical companies and biotech companies that are promising for future drug treatments. And so I'll just tell you a little bit about some of these compounds. So perhaps the most famous are capsaicin, the pungent compound in chili peppers. So I'm sure you've all experienced the heat when you eat these chili peppers. Also here is menthol, the pungent compound in mint that elicits the percept of cooling when you chew mint gum or when you brush your teeth. And also members of the Brassica family produce a wide range of isothiocyanates that are very pungent. And this includes wasabi. And so if last time you ate sushi, you might remember eating some wasabi and getting that very pungent, burning, tingling sensation that goes into your nose. And by studying how these three compounds work, we and others identified a number of molecular transducers that interact with these natural products that then trigger pain sensations. And of course, these plants have evolved to produce these compounds that are irritating, that target the pain pathway, mainly to ward off predators like deer and squirrels that eat seeds and destroy them and block their ability to spread and proliferate. So how do we take these plant products and study their function? One way we do that is to look at the effects that they have on cellular activity. So I'll just show you this experiment here where cells were isolated and placed in a dish. These are sensory neurons that we know mediate these sensations of itch, touch, and pain. We could put them in a dish, and remarkably, in culture, they retain many of the same physiological properties. Some of them respond to noxious heat, some of them respond to cold, and some of them respond to icy cold. And what we can do is we can load them with a dye to monitor their activity. And so we've loaded these cells with a dye that monitors activity. When they're purple, they're not active. Oops, I think I was frozen there for a minute. So when they're purple, these cells are inactive. And when we apply a compound to them or apply cold, what we can see is that in response, to applying a stimulus, in this case capsaicin, you could see that here we identified several cells that are activated by the application of capsaicin. And we could quantify these experiments and you could see in response to a control solution, the cells are not responsive. When we apply capsaicin, you could see that there are 3 cells that are activated. And then in response to heat, we see further and more robust activation of these individual cells in response to noxious heat. And through these experiments, we could identify the molecules and genes that are expressed within these cells that mediate these responses to natural products as well as physical stimuli. For example, when we looked at menthol, it was identified that there is a gene that's called TRPM8 that not only is activated in response to the binding of menthol But also, if we probe activity as a function of temperature, what you could see is that body temperature and room temperature, there's very little activity of this molecule. And only when you reach around 10 degrees, which is right around the temperature that we start to experience cold pain, do you see activity of this molecule. And so by cloning this gene, we could express it in a variety of cell types to study its activity, to show that it's a temperature dependent protein. But we can also then probe its role in a variety of animal models to ask what is its physiological role under normal conditions. And so to do that, what we and many other people do is we could generate transgenic mouse strains where we can delete a specific gene. In this case, we deleted the cold and menthol receptor TRPM8 gene. Otherwise, this is a normal functioning mouse that has no overt defects. And then we could ask specifically, what is the consequence of deleting this gene, in this case, in its ability to sense cold? So I'll just show you an experiment here. Let's see, I'm gonna just stop this briefly and see if I can play this movie. So in this movie, what you'll see is we designed a temperature preference chamber where on one side we could keep the platform that the mouse is on set to 30 degrees C, which is very comfortable, a preferred temperature. And then we could change the temperature in the control plate. So here's a wild type mouse. It has none of its genes deleted. Here it's on a 30-degree platform. Here's 20 degrees, which is probably the temperature inside your house right now. And it's not painful, but what you could clearly see is that this wild type mouse prefers to be at 30 degrees. And even though it's not noxious, it doesn't choose to spend its time here. Now, this is this mouse's brother, where we deleted the TRPM8 gene. And what you can see, even without any quantification, is that this mouse lost its ability to detect this colder temperature here. And we did this at a wide range of temperatures. Even at 0 degrees, this mouse has no aversion to temperature. It's quite remarkable. And it's equally comfortable at a wide range of temperatures that normally mice and humans would not like to be at. And why are we interested in looking at this? This really identified the first cold sensitive, physiologically relevant gene that is very important for temperature preference under normal conditions. And importantly, we were also able to demonstrate that this gene plays a key role in cold hypersensitivity that's associated with a variety of disorders. And one disorder that we are studying in my lab is chemotherapy-induced peripheral neuropathy. This is nerve damage that's caused by the chemotherapy, not the cancer itself. About 60% of patients who undergo chemotherapy suffer from peripheral neuropathy, including a very painful cold hypersensitivity, such that a gentle breeze could feel like a very extreme cold burning sensation. And about 40% of chemotherapy patients have this nerve damage for the rest of their lives. So it's a very debilitating condition that affects quite a number of people. And interestingly, in animal and preclinical models, TRPM8 plays a very important role in chemotherapy-induced neuropathy and cold hypersensitivity. And so there are a number of clinical trials going on right now to look at whether or not drugs that target TRPM8 could help prevent this cold hypersensitivity and the pain associated with it. So this is just one example of the type of pain work that we do in my lab, and how our basic science studies could lead to directly translational studies pursued by other labs, as well as pharmaceutical and biotech companies. Another compound that we've used quite a bit, I mentioned earlier, is wasabi, which is a member of the Brassica family, as well as alliums, which include garlic. They produce a variety of pungent compounds, including allyl isocyanate, which is produced in wasabi and gives you that burning sensation, as well as allicin, which is a compound in garlic. Interestingly, plants in this family, including mustards, ginger, and garlic, these compounds all mimic inflammatory mediators that are produced by the body in response to tissue damage or disease. And really, it was through studying the natural products produced by these plants that really helped identify a new gene, TRPA1, as the target for not only these pungent compounds in these plant products, but produced by the body. And interestingly, TRPA1, we now know, plays a very important role in pain hypersensitivity and allodynia. So normally, looking at this graph from human patient studies, pain sensations are not triggered by stimuli in the innocuous range. Normally, as you increase stimulus intensity, whether it's heat or pressure, no pain is felt until you reach a certain threshold where that stimulus is capable of causing tissue damage. And what happens is, as you then increase in this range, you get higher and higher pain sensation. Under inflammatory conditions, tissue injury, and diseases like chemotherapy-induced peripheral neuropathy, let's see, what happens is that now you experience pain sensations in response to innocuous stimuli, and also in the noxious range, you experience heightened pain sensitivity. And we now know that TRPA1 plays a very key role in this switch where innocuous tactile sensations or temperature sensations trigger pain. Now, inflammatory mediators not only target this TRPA1 gene to trigger pain, but under some conditions, there are different inflammatory mediators that can trigger chronic itch sensations. So I'll just tell you a little bit about that. Maybe some of you have been experiencing itch lately from wearing masks. I know there's a lot of complaints about that, and there's even been some dermatological conditions associated with that. And so itch, as you know, is very irritating. It's defined as an irritating sensation that causes a desire to scratch. And you know, many of you, like these cats and dogs, and this person here with this back scratcher, have come up with clever ways to scratch that itch and alleviate that itch. And there's great pleasure that's associated with that. And these are all examples of acute itch. So mosquito bite, the itchy mask. But I want you to try to imagine what your life might be like if the very worst itch you ever experienced were to spread over extended parts of your body, and if that itch were present for every second of every day. And that's what people who suffer from chronic itch describe. It's a very debilitating condition that I think is very accurately depicted here in these works of art that are based, show depictions of descriptions from Dante's Inferno, where the falsifiers were punished with a burning rage of itch that nothing could relieve. And so Dante chose to punish in his great story the falsifiers, his very worst enemies, with chronic itch. And here you could see them where nothing could relieve that itch. So I think that for chronic itch, Dante has a better definition of what it's like to suffer from chronic itch. Now, 1 in 10 people in their lifetime will suffer from chronic itch, I'm sorry to say. And chronic itch is associated with a variety of disorders. So allergies might come to mind, skin conditions like atopic dermatitis or eczema, but also many other diseases and can cause itch. So kidney and liver failure, some forms of diabetes, infectious diseases, including HIV. And there's a growing list of drugs that have chronic itch as a side effect, including SSRIs and morphine and chloroquine. And the one thing that all of these disorders have in common is that antihistamines are completely ineffective in treating chronic itch. And it's thought that there are different mechanisms in these different disease states that lead to the chronic itch state. And as a result, we know very little about the molecular mechanisms underlying chronic itch. And so my lab and others, and especially people in the pain field, became very interested in understanding mechanisms of chronic itch, because we know that there's a very close interaction between itch and pain. So if you feel itchy and you scratch that itch, you know that scratching alleviates that itchy sensation. And that's because pain can inhibit itch. And normally when you scratch your skin really hard, it hurts. But under chronic itch conditions or even a mosquito bite, that pain feels good. And we also know that there are some cells and molecules that have been implicated in pain that are also involved in itch. And interestingly, if you look at morphine, many patients who are in the hospital and receiving morphine treatment develop really severe chronic itch. That's so severe that and makes them so miserable that they don't want to take the morphine anymore, because they would rather suffer from chronic pain than chronic itch. And these are really severe chronic pain patients who, in some cases, would prefer the pain over the persistent irritating itch that they develop. My lab has become very interested in eczema, which is also known as atopic dermatitis, which is the most common chronic itch disorder. About 20% of children suffer from eczema early in life, but there's also an adult form of eczema. About 10% of people suffer from this. It's characterized by a really red, itchy rash that has certain hot spots throughout the body. And eczema is considered to be an entryway disease for other disorders. So about 70% of patients with severe atopic dermatitis as children go on to develop other atopic diseases later in life, including food allergies, allergic rhinitis, and a very severe form of asthma. And so we postulated that by studying the mechanisms underlying chronic itch, that this would give us insight into how the body is modulated long-term that leads to these other disease states. And I know unfortunately, I had eczema as a kid. It went away, and now I do have asthma. And so in addition to a very important unanswered question in sensory biology and lung biology, I personally am very interested in trying to understand this atopic march, what causes it and how it stops. And what many people don't realize is that in addition to the immune system that contributes to these allergic disorders and atopic diseases, the nervous system plays a very important role in regulating airway activity, as well as mediating those itch sensations in the skin. And interestingly, what we found was that this molecule, TRPA1, and this gene TRPA1, that's not just, that is found in humans and plays a key role in mediating inflammatory pain. We now know that this molecule is activated by inflammatory mediators that are released by immune cells to drive chronic itch and asthma. And here's just a schematic. People probably have heard about mast cells and histamine in the context of a mosquito bite, or perhaps if you've been itchy and you've taken some Benadryl, that acts to alleviate itch through the release of histamine. But under chronic itch conditions, you have many, many different immune cells that release a variety of different inflammatory mediators to trigger itch and allergy, and also asthma. And these diverse inflammatory mediators released by these immune cells under these disease conditions all converge on this TRPA1 ion channel to trigger itch and inflammation in the skin and the lung. And now, very interestingly, neurons as well as the TRPA1 gene is really a hot new target for treating eczema and other atopic diseases like asthma. So I've told you a little bit about how we approach the study of itch and pain, and we are also very interested in our sense of touch and how we experience and distinguish between the brush of a feather and the prick of a pin. And we've known for a long time that there are different subsets of neurons in your body and that innervate your skin that mediate gentle touch and noxious pain. But what we really haven't known, and we still don't really understand this, is how does force excite sensory neurons? How do we convert physical pressure into an electrical signal? How are mechanical pain thresholds set? So, right, that allows our body to distinguish between the brush of a feather or the prick of a pin. And how does injury or disease lead to altered tactile sensitivity, such that in somebody who suffers from chronic pain, the brush of the feather feels incredibly painful. In somebody with diabetic neuropathy, they might step on this pin and not know at all because of the loss of touch sensitivity. And somebody with chronic itch would experience the pain of scratching, that really harsh force, or scratching till they bleed, as pleasurable. And so we're interested in understanding how our tactile system works under normal conditions, as well as these disease conditions, to change this. And I'm sure you've experienced that even just with a mosquito bite, where if you're scratching really hard on one site that's not itchy, you can experience these temporary switches in how we experience a painful stimulus. So to address this, we use a variety of approaches. And one of the approaches that we've used has been to study animals that have unique tactile systems. So what do I mean by that? Well, there's a variety of animals that use their sense of touch to navigate the world. And one of those animals that we've studied is the star-nosed mole. And our idea was that we could study this animal that uses touch as its main sense to navigate the world, as a model to identify mechanically activated transduction molecules in our somatosensory system that mediate touch. Okay, so this is a really unique animal. I'm going to show you a video of this animal in the tunnel system in which it lives. So these moles are functionally blind. They live underground. And what they do is they hunt using a specialized tactile organ called the star. And here is an example of that. So here you can see this animal. It's in this tunnel system. It's probing the surface with this star organ that you see here. And it's looking for small insects that other animals can't detect. So here's a closer look at this animal. You could see this is the star organ. It has these many appendages, about 20 appendages that are independently controlled, that function like fingers. It's surrounding the nose, but it's not important for olfaction, but touch. And its eyes are small, they develop and then they retract, and they're basically covered by hair. So they could detect shadows, but their primary means of navigation is through this tactile organ. And this is actually quite a small animal. It looks big and scary in this picture, but it's about the size of a juvenile rat or a fat mouse. And we became very interested in studying this animal because of its high tactile sensitivity, and also from the work of Ken Catania that showed that this star organ, even though it's the size of your thumbprint, it's innervated 10 times more than the human hand. And I think this video really highlights its high tactile sensitivity. So I'll show you an experiment of that pits a Eastern American mole that doesn't have the specialization against the star-nosed mole. These moles were placed on a glass plate with a camera, a video camera underneath, and then small pieces of worm, their favorite food, was dissected and put on this glass plate. And what you'll see in these videos is these two different moles encountering this food array. So here's our Eastern American mole that does not have a specialized tactile organ. You can see that it licked up a few of the food bites, but for the most part, they're left behind. These are very small pieces of food that we humans cannot detect through sense of touch at all. We could see them though. So now here's the same array. That was shot in real-time video. This is high-speed video. And this is what, oops. And, oh yikes. And here is the star-nosed mole. Already you could see that there's quite a difference. We'll slow this down so it's easy to see what's happening. And so what you can see, these appendages are reaching out. You can see that it touches a food item. And then what happens is there's a second touch from these smaller appendages that are more sensitive right by the mouth. And only after the mole touches something twice, it then decides whether or not to eat an object or a piece of worm in this case. And then you could see its little tongue come out and lick that. And I should tell you that this animal, we had to slow this down because not only is it highly sensitive and able to detect small insects and other larvae in this very competitive ecosystem, the tunnel system that it lives in, where all of these animals live and all eat insects, it's developed this ability to detect small insects that these other animals can't detect at all. And it can detect them very fast. And so in 150 milliseconds, this animal touches something twice, decides whether or not to eat it, and then actually eats it. It's so fast, we can't record the time it takes just to process and decision make. And so it's quite fast. And we thought that this would be a great place to try to identify the molecules that are the genes that are expressed in these particular cells that innervate the tactile organ. So what we did was we were able to identify touch-sensitive neurons from that innervate the star organ. And we were able to identify all of the genes that were expressed and enriched specifically in these highly touch-sensitive neurons. And this led to a very interesting discovery where we identified a receptor that's highly enriched that plays a key role in touch sensation. And I should say that this molecule is not just expressed in this mole, but interestingly, this gene that's enriched in these cells is also enriched in tactile neurons in humans and mice and other mammals. And what we were able to show is that in a wide range of organisms and in cells isolated from humans, that this gene, which is called S1PR3, not very exciting name, but plays a very important role in our ability to detect touch. It turns out that there is a modulator of this gene that we identified, S1P, that's been known to play a key role in inflammatory disease conditions. And under normal conditions, we have normal activity and normal S1P levels and normal activity of this gene that sets pain thresholds. If we silence this gene or we block its activity, it leads to mechanical hyposensitivity that's associated with diseases like peripheral neuropathy, where loss of sensation leads to these repetitive injuries due to mechanical hyposensitivity. And under disease conditions, we know that there's hyperactivity of this gene, and that this leads to mechanical hypersensitivity. So it's kind of like a rheostat where you really need normal activity of this gene to have normal mechanical sensitivity and touch sensitivity. And we know that changing the activity of this gene, either lowering it or raising it, can really lead to a range of different pain disorders. And so this is the last example that I wanted to tell you about from our lab, to just highlight how we approach this. And you really use basic science, studying interesting animals, natural products, as well as a variety of cell-based assays that we use from both human and mouse to really try to identify new drug targets that might lead to new therapeutics for treating human diseases. And a variety of conditions are associated with our somatosensory system that I talked about today, including chronic itch and chronic pain, cancer pain, severe headache. A lot of these painful disorders involve dysfunction of the nervous system, but we still don't understand how this dysfunction is triggered. And finally, in these days of COVID, it's hard not to talk about that. But we now know that SARS-CoV-2, the virus that triggers the COVID-19 disease, can actually infect neurons. And post-infection, a number of neurological disorders are surfacing. And finally, just to bring it back to asthma, we know that these somatosensory neurons play key roles in itch and pain, and I mentioned asthma as well. And we think that they're playing an important role, and that TRPA1 might be an important target in terms of mediating the lung inflammation and airway hyperreactivity that's observed in COVID-19 disease. And these are all avenues that we're currently pursuing. So finally, I just want to introduce you to the lab. These are the people who are in there day in and day out, who are working really hard, even throughout the quarantine on the COVID-19 work. I'm really incredibly lucky to work with such a talented group of students, graduate students, undergraduates, and postdocs, as well as faculty collaborators, who really are the ones who make these discoveries possible. And here is the most recent picture of the lab back from February, when we were able to get together in person. But I just wanted to show you, really who is driving these discoveries in the lab. And also, I'm very fortunate to have funding, both private funding from the Howard Hughes Medical Institute, but also private funding from the National Institute of Health that have really made this work possible. And I just want to thank you for your time today, and coming to hear about our work. Thank you.
All right. Thank you so much, Diana. I think it's time for some audience questions. It looks like they're already starting to flow in. You guys be sure to use the Q&A button at the bottom of the screen if you would like to ask a question. So I guess I'll just start right off here at the top. First question says, do you prefer natural solutions or chemical drugs to treat chronic pain?
Oh, yeah, that's a great question. I think that we have to pursue both. The great thing about natural products, right, is that they're tested. Perhaps we know that they're safe. So for example, capsaicin at small doses, we can use that. And recently there's been a new approval to use high doses of capsaicin as a way to temporarily kill free nerve endings in the cases of cluster headache and other severe pain syndromes. Now, the capsaicin that we eat when we're eating chili peppers, that is not enough. It causes a burning sensation. It does not kill your free nerve endings under normal conditions. These are really high doses where you have to be anesthetized to get them. And so this is a inpatient new treatment that seems very effective for certain types of pain. But then also pharmaceutical companies are very interested in making synthetic derivatives and other compounds. So I think it's great. We could look to natural products and natural remedies, figure out how they work, and see if they could be modified naturally or synthetically as new therapeutics.
Awesome. Thank you so much. Another question here. It says, is there a possible connection between the TRPA1 gene and disorders such as arthritis?
Yeah. So it's thought that this gene TRPA1 encodes this receptor that's very promiscuous. Under normal conditions, it's not active, which makes it a good target for pain, but it seems to be really overactive in a variety of different inflammatory conditions. It pops up in the lung neurons that innervate the lung, under asthma conditions, under arthritis, the nerves that innervate the joints that are inflamed, and the ligaments. Again, TRPA1 is hyperactive there, and also under conditions of tissue injury and cancer-evoked inflammation. And so it seems to be this integrator of different types of inflammatory pathways under different disease conditions. So there's a clinical trial already for asthma, and also it looks pretty good for osteoarthritis.
Very interesting. Thank you. Here's another question. Is there a genetic or molecular cause for notalgia paresthetica?
So there are some genetic causes that lead to peripheral neuropathies, and but in other cases it's not known. So there have been people who have mutations in some of these TRP channel genes that lead to altered pain sensitivity. I didn't talk about any of the targets, but I mentioned that the transmission of the signal is a big target for local anesthetics, and a number of genes have been linked to chronic pain syndromes where people have SNPs in these genes that mediate the transmission of the signal from the periphery to the spinal cord. But there are many, many pain conditions out there where genetic factors or other factors are unknown. But a lot of these pain syndromes run in families. So it doesn't mean that there's not a genetic basis, or it might be multi-genic that causes it. It's just not discovered yet.
All right. Another question says, can there be psychological triggers to chronic itching?
Oh yeah, definitely. So itch is a really debilitating condition. I think people who have suffered from chronic itch or who had eczema as a kid can really relate to that. Um, but it's also definitely um, associated with neuroses. So even just sort of like as a scientist, I'm very neurotic and hypersensitive to many different sensory stimuli, especially thinking about itch and pain all the time. And some of the images that I showed were used in psychophysical experiments and showed that about 60% of people who look at a picture of somebody scratching their arm or see somebody else itching will itch within a few minutes. So if you were itchy, that's normal, but the faster you itch, the more neurotic you are in these psychophysical studies. Um, and there is also a link between, in the past, this idea that if you're scratching all the time, that there's some type of neurological dysfunction or cognitive dysfunction. And interestingly, there was a study in the New York Times a few years ago or there was an article about it. There were all of these children who were institutionalized for self-mutilation, who were then discovered to have a skin disorder that caused them to just scratch constantly. So there's a lot of misinformation about chronic itching. There might be tactile disorders and sensory deficits, certainly that are associated with different disorders. There's been some studies in autistic children who don't like to don't like gentle touch and don't like the feeling of their clothes against their skin. And so I think now there's a lot of interest in studying affective touch and how sensory disorders might be giving rise to some of these things that we consider mental disorders or cognitive dysfunction.
Hmm. Okay, and then what about pleasurable itch? One of the questions says, it seems like my family loves back scratches more than most other people. Is there a role for pleasurable scratching?
Yeah, so I didn't talk about it, but there's been a lot of beautiful work on gentle touch and pleasurable touch, massages, and how that's a very positive part of our life, right? And I think now that we don't have as much human contact, we're really experiencing how important that is. Even if you're, outside when you see somebody, um, and you're 6 feet away and you have your mask on, it's like you normally might embrace somebody, shake their hand. I think we're really suffering emotionally from that lack of physical contact. And I think that really highlights how important this other aspect of touch is in our everyday lives. And there's been some beautiful work identifying a very small but specific subset of these sensory neurons that we and other people work on that are known to mediate that very important social gentle touch.
Yeah, definitely in the times of COVID, touching is not really as permitted nowadays, but hopefully
Right.
we'll get back to a time where we can shake each other's hands and all that good stuff.
Yeah.
Got another question. It says, I was severely affected by psychogenic itch for a few days and then less so over a few weeks. Does any of your research or other research shed light onto psychogenic itch?
Yeah. So there's been a lot of psychophysical studies, but at a molecular and cellular level less. We do know that there is that itch is contagious, not in the case like you get it from touching somebody, but when you see somebody itch or you see a bug, it could trigger these really strong emotional itch sensations. And there's been some nice work looking at neurons in the brain that mediate this response. So just like yawning, if you see somebody yawn, it could trigger you to yawn. We have these mirror neurons that are activated by itch as well. That could be really pretty strong. And this has been shown to occur in animals as well. That a mouse can see another mouse itch, and it's been reported that it will itch, even though it's not really suffering from chronic itch like its neighbor. And so there's a lot of central modulation in our central nervous system that taps into the system. We know that the absence of pain can lead to chronic itch in the case of morphine or other drugs. And so I think there's a lot of potential pathways that might be modulating that triggers these periods of itch that could be really intense, even without a rash. And um, there's even a pretty famous case where somebody had a perceived itch in their head, even though there was nothing there due to neuropathic nerve damage. So lots of potential ways where these types of periods of itching could occur. And I think due to stereotypes about, people who are scratching or who have scabs often gets misdiagnosed as some psychological dysfunction, but it might not be, right?
Yeah, very interesting. Yeah, I have a lot of people here talking about their own cases here. One of them said, I've found that when I have hives or chronic itch that is not relieved by antihistamines, it is often a symptom of low-grade anemia and needs to bump up their iron intake. Do you have any?
Oh,
That,
interesting. Yeah, I don't know about the iron connection. That's very interesting. But it's, yeah, definitely there's a lot of different home treatments for treating itch.
mm-hmm,
Um, so some people like really, really scalding hot water, if you could stand it, but that goes back to this idea that pain inhibits itch. And I prefer ice. So putting on ice, you activate the cold pain pathway a little bit, and that prevents the itch signal from getting to your brain too.
mm-hmm. Awesome. Another question here says, could you please explain the key differences in the neurological responses in hypersensitivity versus desensitization to continuous exposure to stimuli?
Yeah. So that's, yeah, it's a great question. Um, we know that there's sensitization and desensitization that occurs at multiple levels. So, for example, maybe when you put on a sweater, you could feel the mechanical pressure of that against your skin. And what happens is that's activating gentle touch receptors, but then they can rapidly adapt at the level of the free nerve ending so that they're no longer responsive, and you then do not feel like you don't really feel the weight of your clothes after you put them on. You don't really think about it. Although now you will. Sorry. But then there's modulation at multiple levels. And so with prolonged stimulus, you can start to see modulation at the level of the spinal cord as well as at higher brain levels. So under chronic conditions where you now become hypersensitive, what you see overactivity of the peripheral nerves, but then a change that happens at the level of the spinal cord and the brain as well. Under prolonged chronic pain conditions where you now not only affect cellular activity, but you change the cells that are talking to one another in the spinal cord and who's sending the signals up to the brain. And then in people with chronic pain and chronic itch, you can do brain imaging and see that there's more activation of the pain or the itch center and different brain regions that are activated and hyperactive. So we hope that we can try to block in the periphery or block the cause of the pain before these long-term changes happen in the central nervous system that are much harder to then reset.
Interesting. Okay, another question here, a little bit of a long one. It says, I have CIPN. It was worse in the early stages, but subsided to fingertips, toes, and bottoms of my feet. I'm a flutist and wonder if my playing could have had any effect on improving the neuropathy. Also, I used to be able to have acupuncture in my hands and feet, but now it is excruciating. If
Mhm.
I'm numb, why is it more painful?
Yeah, so I didn't get into all of this, but we have maybe, I don't know, 50 to 80 different types of neurons that do different things. And so I'm sorry to hear that there's pain also with numbing and tingling, but those sensations are thought to be mediated by different cells. And so this has been observed where you could have pain hypersensitivity due to damage to the pain neurons, but then the gentle touch neurons might also have some damage as well that triggers these other sensations.
Mhm.
It's true with uh, some forms of autism where there's a hypersensitivity to touch, gentle touch, and altered different loss of pain sensitivity. So you don't always see um, specifically just the pain pathway being targeted, and also the proprioceptors that allow us to have reflexes. Those can be targeted as well.
Great. Um, got a few more questions here. We said one of them says, can you increase S1P to reduce neuropathy?
Um,
Neuropathy. Sorry,
no, that's
neuropathy. I got it.
so we are very interested in the S1P3 gene and also modulating S1P levels. So this gene that we found that seems to be really important in setting pain thresholds. Um, we're looking at its function in CIPN and other forms of neuropathic pain. There are some drugs that are approved by the FDA for other conditions that look quite promising, but they have some off-target effects. And so there's some interest in finding new inhibitors that are really specific for the S1PR3 gene. It's part of a larger gene family, and the drugs that are out there target other family members that play roles in other parts of the body. And so what we want to really do is look at, can we specifically modulate S1PR3 and not the other family members with more specific drugs or natural products to try to treat neuropathy that way.
Great. Great answers, by the way. I'm loving this. I think everyone's enjoying this. Couple-
Questions?
yeah, these are great. Here's a great one too. In terms of current or future medical trials, how is your research planning or being used? Are any of these studies seeming to be very promising for treating chronic pains in a way that current treatments do not?
Yeah, so my lab, we do basic science. We often um, try to identify new genes, molecules that haven't been previously implicated. We primarily work in preclinical models, but fortunately through the wonderful donor system we have in the United States, we're able to get patient samples and human cells where we could, at least in preclinical studies and cellular assays, validate what we find in animal models. And then after that, then it's up to the biotech companies and pharmaceutical companies to decide then, oh, we think that these are convincing preclinical studies, and then take it to the next step. And fortunately, a number of the molecules and genes that we've worked on have been picked up by other companies and carried on and are in clinical trials right now.
Great. It's impactful work then. I think this is our last question before we end it here. It says, are the mechanisms for pain from simple compounds such as formic acid and amphites basically different from those of the complex plant compounds that you mentioned?
So yeah, so I've mentioned plants, but we're also interested in insects and scorpions and stinging ants. So basically, we try to study anything that causes pain or itch, or natural products, including insect venoms and other molecules that alleviate them. So if anybody has interesting compounds or organisms, you could send me an email. And I think that we've learned a lot too, and I didn't have time to talk about it, about like stinging insects and venoms from bees and snail toxins, poison ivy and other plants that cause itch. A lot of the molecules that these plants and animals produce is to ward off predators and prevent them from being killed by us and animals. And these plants, right? Humans like chili peppers and wasabi, but most other mammals don't. They're effective until we came around, at least in protecting the plants and the animals. So I think that and a lot of drugs that are out there, I should state, that are already on the market that are FDA approved were designed based on toxins that came from scorpions and snakes, and that we really need to look at natural products because there's millions of years of evolution that have formed that where these plants and animals have evolved ways to target the pain system as a warning system.
That was a great last question. So I think we'll wrap up here. Thank you guys all for tuning in to Virtual Cafe Sci this evening. And thank you so much, Diana, for taking the time to speak with us today about itch, touch, and pain. I know for me personally, this topic was extremely interesting. And I know based on our chat, our attendees definitely found it fascinating as well. For the audience, thank you guys again for tuning in this evening. Our next Cafe Sci will be coming up in February, so be on the lookout for the registration link in our follow-up email. We will also include a link to the replay for this event. I know a couple of you asked that in the chat, so be on the lookout for that as well as a short survey for you guys to take to help us improve our future events. And with that, have a great rest of your Thursday evening, and I look forward to seeing you guys all next month.
Thanks so much.